Your fingers find them in the shower. The rough patch on the temple that was not there 2 years ago. The small dome on the chest you first noticed at 53. The tag under the arm that catches on clothing. The red dot on the abdomen that appeared without injury. You examine them in the mirror and wonder when each one arrived and why the skin that stayed clear for 30 years started producing them. You assumed aging. You assumed genetics. You assumed the bumps were random, cosmetic, meaningless beyond appearance. Each one has a molecular explanation. Each one traces to a specific system that failed at a specific moment. Each one started as a single cell that divided when it should have been stopped. The physics behind each bump type is different, and the physics explains not just why the bumps appeared, but why they appeared, where they did, when they did, and what each one reveals about the specific quality control system that failed at that specific location. Each bump is a colony. Every visible growth on the skin after 50 began as a single cell that acquired a mutation in a growth-promoting gene and divided when it should have been stopped. The division should have been caught by a quality control protein that checks every cell cycle for DNA damage before allowing the division to proceed. The protein has been checking for 50 years. The protein's own DNA has been accumulating damage from identical ultraviolet exposure and oxidative stress it was built to detect. The inspector is going blind from the conditions on the factory floor. The analogy is precise because the biology is precise. The inspector protein is made from DNA that sits on one factory floor as the DNA it inspects, absorbing identical radiation, accumulating identical damage, losing function at an equal rate as the products it is checking. The inspector and the product are identical material in one environment. The bumps that appeared after 50 are the cells that slipped past an inspector whose capacity to inspect has been declining for decades. What is the inspector, and how does it check each division? P53, the most mutated gene in human cancer, the most studied protein in molecular biology, the checkpoint that guards every cell division in the body. Before a cell divides, P53 protein accumulates inside the nucleus and reads the DNA for damage. The reading is not metaphorical. The P53 tetramer physically binds to specific DNA sequences and scans for structural distortions, mismatches, and strand breaks through the shape of the DNA helix. A damaged section of DNA produces a distorted helix shape that P53 detects through its DNA binding domain, a shape recognition principle antibody paratopes used to detect foreign molecular structures. If the DNA is clean, 53 remains at low concentration and the cell proceeds through division. If the DNA is damaged, P53 concentration rises rapidly, activating two downstream pathways. Pathway 1, cell cycle arrest. P53 activates the gene for P21, a protein that blocks the cyclin-dependent kinases the cell needs to progress from the G1 checkpoint into S phase, the phase where the DNA is copied. The division pauses. The repair machinery has time to fix the damage before the copy is made. If the repair succeeds, P53 concentration drops and the cell proceeds. The pause is temporary, lasting hours to a day while the repair enzymes work. The P21 protein holds the brakes until the repair is confirmed, then degrades, releasing the kinases and allowing the cell cycle to resume. The system is designed for recovery. Most checkpoint activations result in successful repair and continued division. Apoptosis is the fallback when repair cannot restore the DNA to acceptable fidelity. Pathway 2, apoptosis. If the damage is too extensive for repair, P53 activates the genes for Bax and Puma, proteins that perforate the mitochondrial membrane from the inside, releasing cytochrome C into the cytoplasm, triggering the caspase cascade that dismantles the cell from within. The caspases are proteolytic enzymes that cleave the cell content systematically, cutting the structural proteins, fragmenting the DNA, packaging the remnants into membrane-bound vesicles that neighboring cells and macrophages consume. The death is orderly, contained, and leaves no inflammatory debris. The cell is recycled rather than abandoned. Approximately 10 billion cells undergo apoptosis in the human body every day as part of normal tissue maintenance. The process is routine, silent, and invisible. The bumps appear when apoptosis fails because the P53 pathway that triggers it is itself compromised. Programmed death. The cell is destroyed rather than allowed to divide with unrepaired damage. The inspector condemned the product and pulled it from the line. Now the inspector has been running this checkpoint on every division of every cell in the skin for 50 years. The basal keratinocytes in the epidermis divide approximately once every 2 to 4 weeks, renewing the skin surface continuously. Each division is a checkpoint event. Each checkpoint event requires functional p53 protein reading the DNA accurately and activating the correct downstream pathway. The arithmetic is staggering. A single basal keratinocyte divides approximately 600 to 1,500 times across a 70-year lifespan. The skin contains approximately 2 billion basal keratinocytes. The total number of checkpoint events p53 must perform across a lifetime exceeds 1 trillion. 1 trillion quality control assessments by a protein whose own gene degrades with every decade of service. The fatigue is molecular. Each UV photon that strikes the p53 gene has an equal probability of creating a thymine dimer in the inspector as it does in any other gene. The inspector is made from identical material to what it inspects. The factory floor hazard degrades the worker and the product equally. The inspector runs thousands of checkpoint cycles across a lifetime on a factory floor, where the ultraviolet radiation and the oxidative stress are damaging the inspector's own DNA. Inspector accumulates damage from identical exposure it was built to detect. The p53 gene itself is a DNA sequence sitting in the nucleus of every keratinocyte, exposed to the same ultraviolet photons and reactive oxygen species that damage the genes it checks. Mutations accumulate in the p53 gene across decades, progressively reducing the protein's ability to detect damage accurately, to activate p21 for arrest, and to activate Bax and Puma for apoptosis. The checkpoint weakens. Cells that would have been caught at 30 slip through at 60 because the inspector reading the damage report is itself damaged. UV-induced mutation accumulation in p53 and its progressive effect on cell cycle checkpoint function is established molecular biology. Brash documented this in photochemistry and photobiology, confirming that p53 mutations accumulate preferentially in sun-exposed skin through identical UV damage the protein is designed to detect. The accumulation is measurable. Immunohistochemistry studies comparing p53 protein function in young versus old sun-exposed skin show progressive loss of checkpoint activation efficiency, with keratinocytes from older donors requiring more DNA damage to trigger that p53 response that younger cells mount from less damage. The threshold for detection has risen. The inspector needs more evidence to act. Well, the inspector analogy landed differently the week I traced the rough patch on my temple to a checkpoint failure by a protein whose own gene was damaged by identical sunlight that damaged the cell it failed to catch. The inspector and the product on the factory floor are exposed to one hazard. The inspector goes blind from that light that damages what it inspects. The product slips through because the inspector can no longer see it clearly. Inspector checked each division. The inspector degraded over decades. The question is what exactly the ultraviolet photon does to the DNA inside the keratinocyte, the specific chemical event that creates the damage the inspector was supposed to detect. What does a UVB photon do to the DNA inside the cell? 2+2 cycloaddition, the most specific photochemistry in this physics. A UVB photon at approximately 280 to 320 nanometers wavelength is absorbed by two adjacent thymine bases on one DNA strand. The photon's energy drives a covalent bond between the carbon-5 and carbon-6 atoms of the two adjacent thymines, forming a cyclobutane pyrimidine dimer, a CPD. The reaction is photochemical. The UVB photon provides the activation energy for the covalent bond formation between carbons that would not bond under thermal conditions. The reaction occurs in picoseconds, faster than any biological process can prevent it. No enzyme blocks the dimer formation. No antioxidant intercepts the photon. The UVB photon reaches the adjacent thymines and the dimer forms before any protective mechanism can intervene. Prevention is external: sunscreen, clothing, shade. Once the photon arrives at the DNA, the dimer is inevitable. Two bases that were held in position by hydrogen bonding and base stacking interactions are now covalently locked together by a four-membered carbon ring. Dimer distorts the double helix. The normal geometry of B-form DNA requires each base pair to sit at a specific angle and distance from its neighbors. The covalent bond between the two thymines bends the helix by approximately 7 to 9 degrees and unwinds the local twist. The distortion is the damage signal. P53 was built to detect the abnormal geometry where the repair machinery must intervene before the next division copies the error into a permanent mutation. The Sun discussion described UVB photons reaching the basal keratinocytes after penetrating the epidermis. Those photons produce the erythema the skin surface displays as sunburn. The same photons reaching those cells also produce thymine dimers in the nuclear DNA, a damage event that occurs simultaneously with the surface erythema but is invisible from the outside. The two events happen in one cell at one moment from one photon. The surface damage you see as reddening and the code damage you cannot see as a thymine dimer. One photon. Two consequences. One visible, one hidden. The hidden one is the one that matters for the bumps that appeared decades later. The sunburn you see is the surface consequence. The thymine dimer you cannot see is the code-level consequence from one photon. A single hour of midday summer sun produces approximately 100,000 thymine dimers per keratinocyte, 100,000 helical distortions in the DNA of one cell from 1 hour of exposure, each one requiring NER to find it, excise it, and repair it before the next division copies it into permanent mutation. The sheer volume of damage per exposure explains why the repair system falls behind as it ages. The workload is enormous and the capacity is declining. Multiply the per exposure load across every sun exposure, across every year, across every decade. The cumulative thymine dimer count in a sun-exposed keratinocyte by age 60 is any reasonable estimate of what the repair system can clear completely. Some dimers persist. Some persist through the next division. Some become permanent mutations. The mutations that survive repair are not randomly distributed. They cluster at pyrimidine sequences where thymine dimers preferentially form, creating mutation hotspots in the genome that accumulate damage faster than surrounding sequences. The P53 gene contains several of these hotspots, which is why P53 accumulates UV-induced mutations at a rate higher than the genome average. Nucleotide excision repair removes the dimer. The NER pathway detects the helical distortion, excises approximately 24 to 32 nucleotides surrounding the dimer, and resynthesizes the correct sequence using the undamaged complementary strand as a template. The repair takes approximately 30 minutes per dimer in young skin. The repair rate slows measurably with age. Studies comparing NER efficiency in fibroblasts from young and old donors show a 30 to 50% reduction in repair capacity by age 65. The decline reflects decreased expression of the XPC and XPD proteins that recognize and unwind the damaged DNA, accumulated oxidative damage to the repair enzymes themselves, and reduced availability of the NAD+ cofactor the repair pathway requires. The repair machinery ages because the repair machinery is built from proteins, and proteins accumulate damage from one oxidative environment they operate in. Now, the race. Every thymine dimer is a race between repair and replication. If NR removes the dimer before the next cell division, no mutation results. If the cell divides before the dimer is repaired, the replication machinery encounters the dimer and inserts an incorrect base opposite the damaged site, typically an adenine where a cytosine should be. The transition is specific and directional. UV-induced mutations at thymine dimer sites are almost always C to T or CC to TT transitions, a mutational signature so distinctive that genomic analysis can identify UV-induced mutations from their sequence context alone. The signature is the forensic evidence of sun exposure written into the DNA of every keratinocyte that absorbed a UVB photon and failed to repair the resulting dimer before dividing. The error becomes a permanent C to T transition mutation. The mutation is locked into the genome. Every subsequent division copies it faithfully. At 30, the repair wins the race almost every time. Fast NER removing dimers in 30 minutes. Strong 53 catching any cell that reaches the checkpoint with unrepaired damage. Few accumulated mutations to distract the repair machinery. At 60, the repair loses more races, NER now taking 60 to 90 minutes per dimer, P53 requiring more damage to trigger, an accumulated mutation burden that means more damage competing for declining repair capacity. Each lost race adds a permanent mutation to the keratinocyte genome. Some mutations land in growth-promoting genes and drive the clonal expansion that produces the visible colony. How does one mutated cell become a visible bump? Clonal expansion. One keratinocyte acquires a growth-promoting mutation, FGFR3 or PIK3CA, in the case of seborrheic keratosis, the rough, waxy, stuck-on patches that appear on the trunk and face after 40. The mutation activates a growth signaling pathway that increases the cell's division rate relative to its neighbors. The mutated cell divides. Its daughters carry the mutation. The daughters divide. The colony expands. Hafner and colleagues identified these specific mutations in seborrheic keratosis in the Journal of Investigative Dermatology, confirming that FGFR3 activating mutations drive the clonal expansion, producing the characteristic raised, sharply demarcated patches. Expansion is slow. Months to years from the first mutated division to a visible patch. The colony grows because the mutated cell divides faster than its neighbors, gradually displacing the surrounding normal cells, building a patch of genetically identical cells carrying the original growth-promoting mutation. The bump you discovered at 53 began as one mutated cell perhaps 3 to 5 years earlier, growing slowly through the epidermis as the colony expanded from one cell to thousands, each generation carrying the original mutation and each generation dividing faster than the normal cells surrounding it. The growth advantage is modest, perhaps 10 to 20% faster division than the surrounding normal cells, but the advantage compounds over years the way a slightly higher interest rate compounds over decades. Small advantage. Long time. Visible colony. The mathematics of exponential growth applies. A cell dividing 20% faster than its neighbors doubles its population approximately 3.5 times faster. After 1 year, the colony contains cells the surrounding tissue would not have produced for another 3.5 years. After 5 years, the difference is visible as a raised patch containing thousands of cells the normal turnover rate would not have generated. Growth rate is approximately 1 to 2 millimeters per year. The slowness is why the bumps seem to appear suddenly. The colony has been growing for years below the threshold of visual detection, expanding cell by cell at a rate invisible to the eye until the patch crosses approximately 3 to 4 millimeters in diameter and becomes palpable under the fingers. The patch that measures 8 millimeters at discovery has been expanding for approximately 4 to 8 years from the initial mutation event. And then the mutation field. Martin Corena and colleagues measured the somatic mutation burden in normal sun-exposed eyelid skin in Science. The finding, tissue that appears completely normal, clinically unblemished, carries 2 to 6 mutations per megabase of DNA. Notch1 is mutated in approximately 20 to 30% of sun-exposed keratinocytes in apparently normal skin. 20 to 30% of the cells in the clear skin on your face carry a mutation in a growth-regulating gene. The skin looks normal. The genome beneath it does not. The mutation density in some sun-exposed areas approaches the density found in certain cancers, though lacking the specific driver combinations malignancy requires. Bumps are the visible peaks above a mutation landscape covering the entire skin surface. The clear skin between the bumps carries mutations that have not yet landed in the right combination of genes to produce a visible colony. More mutations. Weaker inspector. More combinations. More colonies slipping through, counting 5 skin growths at 50, 15 at 65, and 30 at 75, is counting the output of this exponential divergence between mutation accumulation and quality control capacity. The progression decade by decade is the quality control losing ground against a rising tide of accumulated mutations. At 30, few mutations, strong inspector, rare colonies. At 50, moderate mutations, weakening inspector, occasional colonies. At 70, dense mutation field, severely degraded inspector, multiple colonies visible across the entire sun-exposed surface. The decade-by-decade increase in skin growths is the predictable output of an exponentially growing mutation burden being screened by an exponentially weakening quality control system. My understanding of the mirror shifted the afternoon the mutation field reached my own skin. The seborrheic keratosis on my chest was one colony from one cell whose FGFR3 mutation drove clonal expansion past a p53 checkpoint that was too degraded to catch it. The clear skin beside it carried thousands of mutations per square centimeter that had not yet combined into a growth-promoting configuration. The bumps were the visible minority. The field was the invisible majority. Quality control degraded. The mutations accumulated. Each visible growth on the skin traces to a specific failure in a specific system. The skin tags on the neck and axially trace to a different system entirely, one the body broadcasts through a visible surface marker. What do skin tags reveal about the metabolic state? Skin tags cluster at friction sites, the neck, the axillae, the inframammary folds, in individuals with elevated insulin and IGF-1. The insulin resistance discussion described receptor downregulation driving compensatory hyperinsulinemia. The elevated insulin activates IGF-1 receptor signaling and keratinocytes, priming the cells for proliferation. Mechanical friction at the skin fold provides the local stimulus. Two inputs converging. Systemic growth factor priming from the metabolic state. Local mechanical trigger from the friction site. The tag is the insulin resistance made visible on the skin surface. Skin tags correlate clinically with metabolic syndrome, insulin resistance, and type 2 diabetes across multiple studies. The number of tags correlates with the degree of insulin resistance. More tags, more resistance. If you noticed an increase in skin tags during 1 decade, the fasting glucose crept upward. You are seeing one metabolic shift from 2 different vantage points, one from the blood test, one from the mirror. Tag traces to metabolic signaling. The small red dome that appeared without injury traces to a completely different control system losing its balance. How does the red dome on the skin form? Cherry angioma, a collection of newly formed capillaries in the dermis, appearing progressively from the 30s onward. Angiogenesis, the formation of new blood vessels, is regulated by the balance between pro-growth signals, vascular endothelial growth factor, and anti-growth signals, thrombospondinone. With age, VEGF expression increases while thrombospondin 1 decreases. The balance tips toward growth. New vessels form where they were not needed. Structural failure in existing vessels is one vascular aging pattern. Cherry angiomas represent the opposite failure. Inappropriate construction of new vessels. Both trace to vascular control systems degrading with age, one maintaining existing vessels, the other governing the construction of new ones. Why the growth occurs at scattered specific sites rather than diffusely across the entire surface remains an open question. Local triggers at those specific locations have not been fully characterized. The age-related shift in the VEGF to thrombospondin balance provides the permissive environment. What determines the specific location within that environment is not yet resolved. The honesty matters. The physics explains why the angiogenic balance shifts with age. The physics does not yet explain why the shift produces a vessel cluster at one specific point on the abdomen and not 3 centimeters away. The age-related permissive environment is established. The site selection mechanism is not. One more system, the paradox. A cellular response, DE is designed to prevent dangerous growth, that is, through its own chemistry, promoting it. How does a safety mechanism backfire? When a cell accumulates too much damage but p53 triggers senescence instead of apoptosis, the cell stops dividing permanently. A permanent off switch. The cell can never divide again. The mechanism is a safety system, preventing a damaged cell from replicating its mutations. The cell is arrested, not destroyed. The arrest is permanent. The cell remains alive, metabolically active, but incapable of entering the cell cycle again. The cell occupies space in the tissue, consumes resources, and persists for years or decades. The permanence is what makes the senescent cell accumulation a problem of aging. Each decade adds more senescent cells, and the tissue carries a growing population of arrested cells whose collective secretory output scales with their number. Backfire, senescent cells persist in the tissue for years. They do not divide, but they secrete. The senescence-associated secretory phenotype, SASP, includes inflammatory cytokines, interleukin-6 and interleukin-8, growth factors, VEGF and platelet-derived growth factor, and matrix metalloproteinases that remodel the surrounding tissue. The SASP alters the microenvironment around every senescent cell, driving chronic low-grade inflammation through interleukin-6 and interleukin-8, promoting proliferation in neighboring cells through VEGF and platelet-derived growth factor, and remodeling the extracellular matrix through matrix metalloproteinases that digest the collagen framework, normally constraining cell movement and growth. Cellular senescence and the SASP as a paradoxical contributor to tissue aging is established cell biology. CAMPC established this in the Annual Review of Physiology, confirming that senescent cells promote age-related tissue dysfunction through their secretory output rather than through their own proliferation. Paradox, the safety mechanism that prevented one dangerous cell from dividing, creates a microenvironment that pushes dozens of neighboring cells toward abnormal growth. Senescent cell count increases with age. SASP output increases with density. The tissue becomes progressively more growth permissive, the background against which any single growth mutation is more likely to produce a visible expansion. The tissue at 60 is a different tissue from the tissue at 30. Even where it looks identical, the microenvironment is chemically altered by the senescent secretome, the growth restraints are weakened, and any cell that acquires a growth-promoting mutation enters a landscape that is actively supporting rather than suppressing its expansion. The safety system backfired because the secretory consequences of permanent arrest were not the end of the cell's influence on its neighbors. The senescent cell that stopped dividing to protect the tissue from one dangerous mutation is now, through its SASP output, promoting growth in the 50 to 100 cells within the diffusion radius of its secretory output. Cells that carry their own mutations from that mutation field the damaged inspector failed to catch. The safety mechanism prevented one problem and amplified 50 others. The paradox deepens with age. More senescent cells accumulate, their combined SASP output rises, the tissue environment becomes progressively more growth permissive, and the weakened p53 checkpoint must operate against a biochemical background that is actively promoting the growth it is trying to prevent. Whether any of the growths on your skin should be evaluated, biopsied, monitored, or removed is a clinical determination your dermatologist makes through visual and dermoscopic examination. The physics teaches what each growth type represents at the cellular level. The clinician determines whether any specific growth requires intervention. The bump under your fingertip in the shower was a colony, one cell, one mutation, one checkpoint that should have caught it and could not because the inspector had been degrading from the same exposure for 5 decades. The tag under the arm arrived where insulin-driven growth met friction. The red dome on the chest was angiogenesis running without its brake. The rough patch on the temple was a thymine dimer. The repair system did not remove in time. Each visible growth on the skin after 50 is a specific failure in a specific quality control system in a specific quality control system at a specific location, written in the physics of DNA repair declining with age, cell cycle checkpoints degrading from the same exposure they detect, and growth factor signaling shifting toward permissive growth that held the surface clear for 3 decades and gradually, decade by decade, lost its margin. The inspector is still working. The checkpoint still runs. The repair still operates. Each system is diminished, not absent. The growths that appear after 50 are the fraction that slipped through, the colonies the younger system would have caught. The skin carried the physics silently for 3 decades. The bumps are the physics becoming visible. Each one traceable, each one specific, each one's the output of a system that held for decades and gradually, decade by decade, lost its margin.